In this video lecture, we will talk about transcription and translation. And in the beginning, I will talk about the difference between prokaryotics and eukaryotics in terms of transcription and translation from a general perspective. Then I will talk about the genetic code, what we find that the genetic code is universal between living organisms. Then I will move to talk about how mutations, in particular this type of mutations that are point mutations, can affect the protein structure and function. And finally, at the end, I will give you some examples. So let's start with the introduction. In this semester, I have been talking about the flow of genetic information, and I have focused on three main molecules: DNA, RNA, and proteins, and two main processes: transcription and translation. And if you see, to some degree, the DNA has an important role determining which proteins are going to be translated. So there's a relationship between genotype and phenotype. So depending on the nucleotides that we have, the sequence of nucleotides, you will have different amino acids. And we will talk about that today. In particular, we will talk about these three groups of nucleotides will determine the codons, the three nucleotide of the messenger RNA, and those ones will determine the amino acid that you will get during the translation. However, this is not the entire story. So not all the proteins are expressed in all the tissues at the same time. When these kind of things happen is, for example, when there is cancer, you have a chaotic gene expression when every gene is expressing at the same time. So what we need is a coordinate process of gene expression. And gene expression has two phases, one that is transcription and the other one that is translation. And we will talk about that today. So let's recap. First, I told you that the information content of the DNA is in the form of specific nucleotides. So if you remember, I talked about that in particular, three nucleotides will determine the three nucleotides of the messenger RNA, that they will be the codons, and we will talk in more detail about that in this class, and those will determine which amino acid will be translated. Second thing that I want that you remember from this slide is that the DNA inherited by an organism lead to a specific trait by dictating the synthesis of a protein. So again, there is a relationship between genotype and phenotype, and the genes are going to some degree determine the protein, the amino acid that you will have. And finally, at the end, I told you about a process, the process by which the DNA directs the synthesis include two stages, transcription and the other one that is translation. That are the processes that we are going to talk in more detail today. Now we're going to start talking about the basic principles of transcription and translation. And here we have a diagram of a prokaryote, a bacterial cell, and a eukaryote. And as we talked before, there's a big difference between prokaryotes and eukaryotes, and is this, the presence of a nucleus. And as a result, transcription takes place in the cytoplasm in bacteria, as well as translation. Both will take place in the cytoplasm. On the other hand, in eukaryotic, the transcription and the beginning of the RNA processing will take place inside of the nucleus, while the translation will take place in the cytoplasm. However, in both cases, the RNA will be the intermediate between DNA and proteins that you see here, a polypeptide. So to summarize, to talk about the basic principle of transcription and translation, I told you that the RNA is an intermediate between genes and proteins for which they code, and that the transcription is the synthesis of RNA under the direction of DNA. In this diagram, I want to show you that the transcription produces as a result messenger RNA that could be in eukaryotic later will be processed, and you can read more about that in your textbook. And also that the translation that you see here is the synthesis of a polypeptide and is under the direction of the messenger RNA. And finally here you can see the ribosomes, and the ribosomes are the sites of translation. So first thing that I show you is that in the nucleus, transcription produces messenger RNA. Then I show you that translation is the synthesis of a polypeptide and occur under the direction of the messenger RNA. And finally, that ribosomes are the site of the translation. Now let's go back again in this schematic of the prokaryotic cell. And I already told you that as they don't have nucleus, these two processes occur together. And this has as a consequence that the messenger RNA, if you see here, there is no processing. So there is not a processing, and we will talk about the processing of RNA, and in your textbook they describe it very well. So first that you have to remember of this slide that the transcription and translation occur together in the cytoplasm. And the second thing that I want that you remember is that in prokaryotics, the messenger RNA produced by transcription is immediately translated without more processing. On the other hand, in eukaryotics, you have the nucleus, and if you see here, there are gaps, they are called nucleopores, and the messenger RNA cross through this complex of proteins, and they can go to the cytoplasm, and then can go to translation. However, before this takes place, you have a pre-messenger RNA, and later, if this is processed, there is an RNA processing that will give a cap and a tail to the RNA. And if you see here, it will become smaller. So it will remove some sections of this pre-messenger RNA. So to recap, in eukaryotics, different from prokaryotics, RNA transcripts are modified before becoming a true messenger RNA. And I show you that the processing will include to have a cap, to have a tail, to make them smaller, and we will talk about that later. In eukaryotic cells, the nuclear envelope separates the transcription and translation, as we previously talked. And finally, in eukaryotic, RNA transcripts are modified through RNA processing to yield the finished messenger RNA. This pre-messenger RNA is also known as primary transcript and is the initial RNA transcript of any gene before the RNA processing. And we have been talking about DNA, RNA, and proteins. And independent if they are eukaryotic or prokaryotic, cells seem to be governed by a cellular change of command. And this is known as a central dogma. So remember, the central dogma is the concept that the cells are governed by a cellular change of command that goes from DNA to proteins in this direction.